organizers for the efforts they put into this meeting. And I'm going to now share my screen. Okay, everybody's ready. So I'm going to talk about experiments of selection in antibody libraries, which correspond to the PhD work of Stephen Schultz that we co-supervised together with Olivier. It was the follow-up of work of a previous student, Sébastien Boyer. And it's also in collaboration with Simona, Rémi, and Matteo, who are also speaking at this meeting, and I think in the audience right now. So please correct me on the fly if I make a mistake. The work is essentially described in a preprint and will soon appear in the journal. So the big question we're after is a property of proteins that Olivier mentioned in his talk, which is evolvability. And to talk about evolvability, as you will see in a minute, you need to have two evolutionary timescales. And we study this property using antibodies, and I will explain now why we use antibodies. And before that, I will apologize to the immunology experts in the audience for this very schematic view of the antibody response. So you can view an antibody repertoire as a huge collection of proteins, which are typically 100 amino acid long. And there's a lot of sequences where there's very low diversity on along this sequence of 100 amino acids. Most of the diversity, which is very high, is focused at very few sites, which belong to a small segment, which is called CDR3, and towards the end of the sequence, towards the C-terminus. And the number of different CDR3 loops that you can have in a repertoire is on the order of 1 billion, and can be bigger or smaller depending on the organism and the species and the immunological status, but that's typically a number you will find. So these repertoires are formed through a process of recombination that I'm not going to explain, starting from genes in your genome that generates this very high diversity. I just want to mention that one point that we will use, and you should remember, is the fact that only the low diversity part of the sequence of antibodies is actually fully encoded in the genome. Most of the CDR3 sequence, which is highly diverse, is not encoded in the genome. It is essentially a random. And so the immune response starts through a selection step, during which antibodies with the highest affinities to the antigen that you are encountering get selected. And to a large extent, the binding affinity and binding specificity of antibodies to their antigen is thought to depend mostly on the CDR3 loop. And you can understand why, simply because the CDR3 region corresponds to a small loop that sticks out from the antibody structure and directly contacts the antigen. So it's clearly responsible for binding. Quantitatively speaking, the number of different CDR3 loops is thought to control the highest possible affinity that you can get from a repertoire. Okay, that's the one quantitative aspect that is more or less understood. Now, the second step in the immune response is a process, it's a Darwinian process called affinity maturation. It's a process during which the sequences of antibodies that were selected in the first place get randomly mutated all along their length in the CDR3, as well as in the low diversity segment, in the low diversity part of the sequence. And the antibody mutants that have the highest affinity gets selected further, and several rounds of this process produce very high affinity antibodies in a couple of days or weeks. So this is a quite fast process. And now the last point is to not forget the fact that the only genes that are transmitted to the next generation in the next organisms are the germline genes, which are the genes that are used to produce the antibody repertoire. But the sequences of antibodies that are selected or affinity maturated are not transmitted to the next generation. There is no Lamarckism in a way. So we talk about evolvability of germline genes of antibodies because they evolve on the long time scale of evolution to be able to evolve on a fast time scale of the affinity maturation, and in particular of this Darwinian process of affinity maturation. So this is why we talk about antibody scaffold evolvability. I forgot to say we call scaffold precisely the region of the antibody which is of low diversity and whose sequence is germline encoded and gets transmitted to the next generation. Now, to correct the picture that I just gave you, there is one more ingredient, is the fact that I've been talking about low diversity, but it's non-zero diversity. There is significant diversity all along the sequence of the antibodies, and that's because the germline genes encoding antibodies are quite diverse. We've got tens of alleles of these genes in our genome, and essentially we have a diversity at the level of the scaffold that is such that the repertoire is composed of sub-repertoires, each one relying on a different antibody scaffold, bearing the same amount of diversity, roughly the same amount of diversity in the CDR3 loop. So in a way, these sub-repertoires based on different scaffolds compete with each other during the selection step and also during the affinity maturation step. So I already mentioned this. We have a scaffold sequence which gets inherited by the next generation and not the CDR3 sequence. We have different scaffolds in our repertoires. Do they correspond to different selection potentials? What I mean by that is they all bear more or less the same CDR3 diversity at the genetic level, but does this translate into differences in terms of phenotypic diversities of these different sub-repertoires? Finally, the long-term question is to understand whether these different scaffolds correspond to different levels of evolvability, and this is not the object of today's talk. We're going to focus just on this first question about selection potentials. So now we'll perform experiments to analyze this. And how do we go about it? So we produce antibody sequences in vitro based on different scaffolds that we pick in databases, for example. And we randomize 4 positions in the CDR3 loop. We randomize them exactly the same way in all of our libraries, and 4 different positions are allowed to vary over the 20 amino acids, so that's roughly 100,000 sequences in each of our libraries. And then we take all of these sequences together, so say we have different libraries mixed together in the same tube, so several 100,000 sequences in the same tube. And then we put them in a phage display format, which is a molecular biology technique that allows you to select antibodies for binding to some arbitrary target that you immobilize on magnetic beads, and then to recover the sequences of antibodies that bound specifically and tightly to the target. Then we take these sequences and we amplify them in E. coli to be able to cycle this process, and we typically need to cycle this 2, 3, 4 times. The reason is, especially the binding step is noisy, and also the amplification step, so we need to cycle this to get rid of the noise. And then to monitor what's going on, what we do is we deep sequence the population of antibodies, the meta library of all these antibodies at different steps, and in particular, just before and just after the binding step, which allows us to determine the frequencies of every clone in the before and after selection, and to compute enrichments of all these clones. And then what we call the response to selection of a library is the distribution of these enrichments. And as I explained, we would like to quantify the selective potential of a library, which is basically expressed by this distribution. And so to be able to quantify it with just one number, we would like to be able to model with very few parameters those distributions. And as I will explain now, what we find, and I will detail it in the next slides, we find that the shape of these distributions and the value of these parameters depend on one particular property of the scaffold, which is its maturation degree, which basically means the number of somatic mutations it has accumulated during an affinity maturation process. Sorry, so here are the 3 libraries that we have used in our experiments. They are based on 3 different antibody scaffolds. We start from one of them, which is a germline encoded scaffold, which means a naive antibody scaffold, which has not accumulated any somatic mutations. And we're going to compare it to 2 antibodies that have maturated against HIV, just because we have so many antibody sequences in the databases for this virus. And so these 2 maturated antibodies originate from the same germline antibody. So we have 3 different scaffolds that vary essentially by the number of somatic mutations they have. One is without any somatic mutation, one has 15, the other one 35 out of 100 amino acids. So that's a gradient of 50 maturation of the scaffolds we use in our experiments. These are the sequences of these scaffolds. You don't need to look at them at all. You will just notice, if you have time to notice, that you have an increasing number of mutations when you go from the germline-based scaffold on the bottom to the BNAB scaffold on top. And you will also see that we have 4 positions in the CDR3 loops that are fully randomized. And now in our experiments, what we do is we take these 3 libraries mixed together, and we select them for binding against 4 different targets that have absolutely nothing to do with the original target to which those antibodies had been selected, which are quite diverse. We have 2 pieces of DNA and 2 fluorescent proteins. Now, what I'm showing you is the typical picture of the enrichment distributions of these 3 libraries, which are colored in blue, green, and red here, against, for example, 1 target. And what you notice immediately when you look at this distribution of enrichments is that the germline-based scaffold library yields a much broader distribution of enrichments. The black lines are fits that I'm going to describe in the next slides. If you look at the 4 other targets, it's the same story. You find that the germline-based scaffold yields a much broader distribution of enrichments. There's no data here, that's why there's no plot. So I think by eye it's quite obvious there is an interesting effect, and we would like to be able to quantify it, and this is something that I'm going to present in 2 slides. Before that, well, we can also look at the sequence pattern at these CDR3 positions that you can describe with a sequence logo of the population round of selection after round of selection. So you start from a relatively random population, a random, sequence of CDR3. And you see that as you select for binding, you start to select for a motif at these 4 different sites. And one way to represent this effect that does not depend on the particular round of selection that you're looking at is to compute a sequence logo of the enrichments. Remember, enrichments are the ratios of frequencies at successive rounds. So this picture does not depend on the particular round at which you're looking. So now I'm looking at the sequence logos of enrichments of the germline-based library for the 4 different targets that we had in our experiments. What you see are 4 distinct motifs that get selected. These motifs are different for the different targets. You will notice that they are slightly similar when the targets are similar, and they are much more different when the targets are nothing to do with each other, between the DNA and the protein targets. If you look at the 2 other libraries, especially the one that comes from the most maturated scaffold, you don't have so much of a motif that emerges. The one that is in between in terms of maturation degree yields an intermediate picture. Okay, sometimes you don't have any motif that really comes up. Sometimes you do have a clear motif, and it's actually the same as the germline-based scaffold library for the same target. Now let's stop this qualitative description and try to be a bit more quantitative. And so, as I said, we would like to quantify this selection potential using parameters from a very simple model. And the very simple model that we came up with is this log normal distribution that is shown here, that has only 2 parameters. It has mu and sigma, which represent respectively roughly the average enrichment of your library and the variance of the enrichment distribution of your library. And so we use this model to fit all the distribution that I showed in a previous slide. And there are many reasons why we picked this model, and one of them is that it's consistent with the idea that the 4 different sites of the CDR3 will contribute independently to binding. So now I'm using this, this model to fit all the data that I showed in the previous slide. So we can summarize everything in only one plot, sigma versus mu. Different colors correspond to the different libraries and different symbols to different targets. Some of them, as you see, are in duplicates for duplicate experiments. And what you see is something that you could already see by eye on my previous slide, is the fact that the germline-based scaffold yields a higher sigma, a higher variance of these enrichment distribution. So this is a way to quantify something that we could already see by eye. But what is quite impressive in a way is that even the lowest value of sigma for the germline-based scaffold is above the highest value of sigma for any other library against any other target. So there seems to be a, let's say, a systematic result here. And we would like to propose that sigma is a good way to quantify the selective potential of a library against a particular target during a selection experiment. You will also notice that there's an effect also on the mu axis. For example, the BNAB scaffold library seems to be with a larger mu than the 2 other libraries. And there are several reasons for that. And one of them is that we noticed a posteriori that there's a little bit of binding affinity. It's weak, but there's a bit of binding affinity of the BNAB scaffold itself to the magnetic beads that we use in our experiments. So it's a minor effect, and it's of course much less interesting than this sigma parameter. So schematically, you could summarize this way, with a smaller mu for the germline-based library and the larger mu for the BNAB library, and a larger variance for the blue distribution and the smaller variance for the red distribution. And the net result is that most of the germline-based scaffold library clones are globally the worst antibodies, and the top clones of that library are globally the best antibodies because the sigma is larger. So now you could think of other ways to model this data. And this is actually coming from an idea that we already applied in a previous paper. You can apply extreme value theory modeling using the generalized Pareto distribution. And kappa is the shape parameter of that distribution. And I'm not going to go into detail unless there's questions that I can answer later. But you see that kappa also nicely discriminates between the germline-based scaffold library and the 2 others. So basically, discrimination does not depend on the particular model that you like to see the clear difference you have between the germline-based scaffold library and the 2 others. And finally, the last result I will show is that one thing you can do with this very simple log normal model is predict, at least qualitatively, what happens to the mixture of the 3 libraries. So here you're looking at solid lines at the evolution of the composition of the population of the meta library of the 3 mixed libraries of all the clones, round of selection after round of selection. And what you see is that in the first rounds of selection, the BNAB library dominates the population. And later on, it's the germline-based scaffold library that takes over the meta population. And this you can easily understand because the mu value of the BNAB library is higher, so it's originally advantaged, whereas the sigma value of the germline-based library will make it win if you wait long enough, if you repeat selection cycles long enough. What you see is that the log normal model can nicely reproduce these trends. There's a little bit of a gap between the data and the model, and that's essentially because to fit the model, the hypothesis that was made is that all the clones of all the 3 libraries are at equal frequencies initially, which is not true, and you saw it in the sequence logos I showed you, but we didn't have enough information to fit that in. And that probably explains the gap you see. So in summary, what I told you is that we can model using a log normal distribution the enrichment distributions of our experiments. We propose that the sigma of this log normal model is a nice way to quantify with just one number the selection potential of a library. There are other ways to describe these enrichment distributions that are also yield a consistent discrimination between the high selective potential versus low selective potential libraries. And biologically speaking, we find that the library that is built on the germline-based scaffold has the highest selective potential. In other words, when you take maturated scaffolds, you decrease the selection potential of your library. One result that I didn't show is that if you take the top binders of library that has a high sigma, then those top binders also bind more specifically to their target, which means that binding specificity seems also to depend on the degree of maturation of the scaffold. Finally, some outlook. So on the modeling side, you could think that you can come up with a more precise model. So our model was quite rough. In particular, we did not take into account some noise and biases that we have in our experiments. But once we have a good model, we can think of turning it generative to start to propose some antibody sequences that we could test in the test tube. And finally, some open questions. First of all, is what we report really general? So we are right now in the lab trying to produce maturation trajectories to obtain new scaffolds to repeat the same analysis and to see how general our conclusions are. And another very interesting question is, what are the physical properties that account for differences in terms of selective potential or evolvability later on? And finally, how are these properties encoded in the scaffold sequences? And with this, I will stop and take your questions.
Okay, thanks a lot for this great talk. So people, if you have questions, you can ask them directly or type them in the chat and I would read them. So who wants to start? Okay, I have a question in terms of evolution, because you started the talk saying, I mean, there is this long-term evolution. What do we know a bit like, I don't know, is there a difference between population in terms in terms of this germline library? I mean, that are really different by population, or it's really mixed, or?
So evolution of antibody genes is a difficult topic. So first of all, we have many alleles of antibody genes. So this is one first problem to understand why so many alleles. People have talked about balanced selection of antibody alleles. So there is a diversity intra-organism that is clearly maintained throughout evolution. I don't think it's very well understood. So one of the possible explanation is this effect that we would like to study, evolvability. You need to probably have a diversity of antibody genes to basically be able to respond to many different antigens during your lifetime. I don't know of any good explanation of the diversity we have intra-organism and the evolution of this diversity.
Okay. And what it was interesting. So Martin, you want to ask a question? Go ahead.
One question, because I don't remember well enough the plots you showed in the beginning before you had the log normal model. So the log normal model lives a bit from the fact that independently of the mean of the mu, if you have a larger sigma, at the end you will the larger sigma will win.
Yes.
Do you see this in the data that you the germline always has also the best fitnesses, or?
Yes. So this is more or less what I showed, for example, on this slide.
Yeah.
Okay. Sorry. Can you see it now? Okay. So the germline library that has the highest sigma always wins.
Okay, so that's after the selections you get the best. Yeah.
You need,
okay.
If you repeat those selection cycles, you find that it always takes over.
I was thinking in terms of the plots where you, in the beginning, the effects, but you sometimes saw and sometimes probably not in.
Sorry, here you go.
Yeah.
Yeah, they always win.
Okay.
Okay.
Yeah. Okay. Yeah, that's what I wanted to see. I didn't remember well. I didn't look to the extreme points.
The top germline clones are always overall the top clones.
Okay, thanks.
Okay, so I had a question in the experiment. So you have you compare to you have 4 targets that are two that are proteins and 2 that are DNA.
Mm-hmm.
And do you see do you see patterns? I mean, that are similar for the 2 proteins, similar for the 2 DNA, or is it?
Yeah, this is what you see here. So only this germline base scaffold gives you motifs systematically. You see that for the 2 DNA targets, there's a clear enrichment of blue amino acids, which are positively charged. Remember, DNA is negatively charged, would make sense. It's not as clear for DNA 2 as for DNA 1, but it's significantly statistically there. For the 2 proteins, they are quite different, but structurally they have similarities. Yeah, you see GLL. Okay, you don't have the second amino acid that is identical, but the first number 1, 3, and 4 are identical in the motifs. So you do have similarities in the motifs when the targets are similar. I forgot to say there's a link also between the, let's say, the size of these letters, which is information, okay, that you get. It's relative entropy, and there's an information theoretical argument to say that there's a link between the sigma values and the size of these letters, which is also explained in the preprint.
Okay, there is a question by Lucas Esta. Instead of taking different mutated scaffold libraries, what about performing mutations on the go with the selection process?
So I'm not sure I completely understand the question. So of course we compared scaffolds that bear different levels of maturation. We could have picked scaffolds that differ by other things, okay? So why not take scaffolds that differ by mutations that are accumulated another way? Yeah, that's, why not? We could have taken scaffolds that differ by, I don't know, thermal stability, or there are many different things that we could test also in this regard.
I mean, maybe it's me that I not understand clearly, because you mentioned at the beginning that you perform mutation at the scaffold structure after a selection step. But then what I understand is that you take the libraries of the scaffold and then you perform kind of deep mutational scan experiment without performing further mutations.
Also, in our experiments, we don't do any mutation at all during the process. We just start from libraries that have this diversity in the CDR3 region, and then we just select. We don't introduce any mutation at all. And so maybe to be more precise, I think I understood what you asked. We take 3 different scaffolds that we find in databases, okay? We just know that the 2 top scaffolds are derived from the bottom one. They just differ by somatic mutations. We introduce random mutation there, and then we perform the selection. We don't introduce any further mutation at all. If that's, because you asked me at which point we introduce the somatic mutation in the scaffolds. It's before we start the experiments. We don't do any mutation during the experiment.
Okay, okay, it's clear. And so my question was if you think it would be of any advantage to perform further mutations together with selection.
So yes, and that's to essentially answer this question. So we just looked at differences in terms of response to the selection step of the different scaffolds. Okay, we just look at this step so far, but we would like to study also the differences between scaffolds in terms of response to affinity maturation. And for this, we will have to introduce random mutations between rounds of selection. So that would be, yeah, this is why.
So you are aiming to do that.
Yeah, exactly. Yeah.
Okay, nice. Thank you very much.
Thank you.
Okay, so I think we have to stop the session here. So thanks for all the speakers of the session. Please, there is after there is a gather if you want to talk in a smaller group.